research professor at NOAA's Atlantic Oceanographic and Meteorological Laboratory in Miami. Via a NOAA, so the National Oceanic and Atmospheric Administration, Cooperative Institute and, with Mississippi State University. Luke received his bachelor's degree at Stanford University and his PhD at MIT, both in biology. His current research focuses on marine systems from microbes to fish to mammals using omics methods, especially DNA sequencing. His lab is developing methods to facilitate high throughput environmental DNA sample processing and data analysis, and applying them to monitoring and conservation efforts in the Atlantic Ocean, Gulf of Mexico, as well as the Great Lakes. So take it away, Luke.
Thanks, Rob. Can you guys see that okay? Yeah, great. So yeah, thanks for the invitation to come. It's too bad I couldn't come in person this time, but next time I would love to make it up to East Lansing. I'm originally from Indiana, Big Ten state, and I've spent a lot of time in Michigan, so it'd be fun to come back. So yeah, as Rob said, I'm at NOAA in Miami. The lab is called the Atlantic Oceanographic and Meteorological Laboratory, and I'll tell you a little bit about that at the beginning here. And so yeah, let's get started. So today I want to well, here's the outline of my talk. So first I'll give some introduction about NOAA and AOML, since it's kind of a different sort of research institution from the academic model. And then I'll give an introduction to environmental DNA, or eDNA. I'll talk about some of our work with methods development, lab tools, and then bioinformatics tools. And then I'll talk about the applications of some of those tools in different environments. So more local to us, so South Florida, Gulf of Mexico, and then a little bit further afield in the Great Lakes, actually Lake Erie, not too far from you guys. And then I'll talk about some work we have planned to do global eDNA studies. I'll talk about a study we have looking at methane ice worms, which are these worms that live in the bottom of the Gulf of Mexico. And then I'll talk about also some planning for coral reef projects. So first off, a little bit about NOAA and AOML. So NOAA is a federal agency in the Department of Commerce, and the mission statement briefly is science, service, and stewardship. So science first, but we serve the nation, and we act also as a steward of the environment. So we want to understand and predict changes in climate, weather, oceans, and coasts, share that knowledge and information with others, and conserve and manage coastal and marine ecosystems and resources. So in this image, you can see a really good picture of all the kind of stuff that NOAA does. So I work on these white ships, these NOAA ships, and sometimes some of us get to go scuba diving and study coral reefs and other communities in the water. But we also have the National Weather Service. We have satellites. We just launched a new satellite a few weeks ago. We have planes. So if you've ever seen the hurricane hunters, the people that go in planes and fly into hurricanes, those are actually people based at our lab in Miami that do that. So we have a really broad suite of science that we do. And kind of on the more cutting edge, we've got some recently rolled out science and technology focus areas. So these are the ones, in fact, they all really, I'll talk about all these in some way or another today. We are working in uncrewed systems. So these are ways of sampling the environment that don't require a human. They can, drones and gliders and things like that. Omics, or genomics and the related family of tools, is a big part of our focus. Big data and data management, storage, sharing is a focus. Cloud computing, and then artificial intelligence. I won't talk too much actually about our cloud and AI work today, but that's definitely a big focus too. So AOML is in Miami. This is a skyline of Miami in the background, and then AOML is here in the foreground on the right, and it's a really beautiful location on a place called Virginia Key. And then across the street is the University of Miami's Rosenstiel School of Marine and Atmospheric Studies. So we have a lot of interaction with University of Miami. So in fact, around half of the employees at AOML are actually employees of the University of Miami through a cooperative agreement. And then on the left here, it's kind of cut off, is the Miami Seaquarium, which is the local aquarium. So we're really close to the environments that we study. And then taking a big step back, thinking about where we're located on the Earth. So in Miami, we're kind of right in between the Gulf of Mexico, the Caribbean, and the Atlantic Ocean. And as I mentioned, we even do a little bit of work in the Great Lakes. So we're kind of in a really strategic location to study all these different environments and systems. And then zooming in a little bit, you can see kind of where we are located relative to the Gulf of Mexico and to the Atlantic and the Caribbean, and the Florida Keys are down here. Can you guys see my cursor, by the way?
Not really.
Okay.
Oh, yeah, faintly, yeah.
All right, yeah. Anyway, so you know where the Florida Keys are, though. They're this little branch of islands that comes off of Miami. So we have studies in all these different locations. So moving on, I wanted to give an introduction to environmental DNA. So a good way to think about environmental DNA is imagine your dog is sitting on your car, on the seat of your car, and it's making a mess. And when it's gone, there's still traces left. There's hair, there's some cells, there's things you can't see. And so environmental DNA is the traces left behind from organisms. And in fact, we broaden that definition often to include the microbiome and the different microorganisms that are in an environment as well. So on the left here is showing that one of our ROVs that can go through the water and sample. There's many different ways we have of sampling seawater. And on the right are kind of depicting different processes that might affect environmental DNA. But suffice it to say, there's lots of DNA left behind from fishes, from mammals, from invertebrates, plankton, and bacteria and viruses that are in seawater. And so one of the ways that we study environmental DNA, in fact, the predominant way, is using omics methods. So this could either be different kinds of sequencing, whether metagenomic sequencing or targeted metabarcoding or amplicon sequencing, or it could be quantitative PCR, which is a more targeted method still. And so from all this DNA in the environment, we can really get a picture of the whole community, from microbes to vertebrates and everything in between. And so a typical workflow that we would use, this particular diagram was taken from a paper where they were focused more on a freshwater system like a stream or a lake, but the principles are the same. You collect your sample, and in fact, I'll show some sampling collection devices that we use in the ocean in a minute. You collect your sample, you extract the DNA, you do perhaps a metabarcoding or amplicon sequencing study shown on the right here, then perform some kind of next generation sequencing, and then do a bioinformatic analysis. And hopefully you have something more powerful than this 1990s laptop, but coffee is important, so they got that part right. So moving on now, so I've given you some background about where we are and kind of what eDNA is. So let's talk about some of the tools we use to study environmental DNA, both in the lab and then after that, some bioinformatics tools. So one of the new methods we're using to collect eDNA is this device that was developed at our lab, not so much by myself personally, although I helped test it, but by Ian Enochs and Nate Vermel. And it's a robotic device that can be programmed to filter a preset amount of water at a preset time, and then pump preservative into that filter. So shown here are 5 of these devices just in the lab, but these would normally be submerged a piling or a reef or in an underwater environment. So on the right here, we're testing these by dunking them in the water and then setting them to go off. So that's one way on the lower right here, one way we can get the water. The more traditional way would be via a Niskin bottle or a Niskin bottle on a CTD. So that's what's shown on the left here. This is a bottle that can be dropped down into the water and then electronically or mechanically triggered to close at a set depth. And then you have water that was collected at that depth and you bring it up to the surface, transfer it into a bottle and then filter it. And so after filtering that seawater, you're left with these filters. This is a really common type that we use called Sterivex. It's a little plastic cartridge. And so this is a really nice way to filter because it's clean. The filter is enclosed inside a plastic housing, so it's not exposed to the environment and it's easier to work with without contaminating. The downside is that it's tricky to extract the DNA from this filter. So the old way we used to do this, and many people still do this, is they will take the filter and get a pipe cutter, cut it open or break it open somehow, and then mechanically with a scalpel, cut the filter off. This is very tedious. If you're needing to do hundreds of these, you can imagine it can take a long time and can be potentially prone to contamination. So there's other methods that have been developed to do this inside the filter. And these have been around for a little while, but there's a new variation on this, which is to add garnet beads or kind of like a glass bead into the filter and to do a sort of vortexing, sort of like bead beating, to mechanically free any kind of cellular material from the filter before doing the extraction. And so that's something we've developed, and I'm just going to show you quickly. In fact, I might just skip over this video because I think it's going to be won't render so well. But in any case, we use this robot called the Kingfisher that allows us to do. It's a bead handling robot, and this metal part can go up and down to mix, and then it can be magnetized to transfer the beads from one plate to the next and do all the steps in a DNA extraction, but in a plate using a magnetic arm. And so what we did in our lab was we tested these different methods, different variations on a method where we're either not using any beads or using different kinds of beads with the lysis buffer of different sizes, 0.1 or 0.5 micron or both. Doing the extraction then of that lysate in the Kingfisher Flex robot, and then doing sequencing of either the 16S rRNA gene, which is bacteria, or the 12S rRNA gene of the mitochondrial genome of fish. And I'll show some of this data a little bit later. But at the sort of technical level, we found that all the different treatments gave good concentrations of bulk eDNA. And in the bacteria, there wasn't a lot of difference. However, if you looked at the chloroplast sequences from the bacterial sequencing, so because chloroplasts and bacteria share an evolutionary history, when we use primers for one, they target chloroplasts also. So we found that when we used beads, we were able to get better detection of diatoms and other groups that have a hard casing. And so that suggests that the beads were helpful for that. In addition, for the fish DNA, we got better recovery of the fish sequences when we had either the large beads only or large and small beads together. So we concluded that it does really help to have the beads in there, and we're able to get good data from that. And I'll share some of the data a little bit later. And then next up, I wanted to just cover some of the bioinformatics tools that our lab is developing. So one is called Tourmaline, and this is named after a surf beach in San Diego because it's a pipeline. And so there's another pipeline called Bonsai, which is named after a surf break in Hawaii. So we wanted to pay homage to that and to San Diego, where this work was started. And so this is a an amplicon sequence processing workflow. So we're taking metabarcoding or amplicon data, the kind of data you would work with in Chime, if you've ever heard of Chime. And in fact, this workflow uses Chime 2 at its core, and then adds to it a lot of additional features, including the workflow structure using SnakeMake. It provides an HTML report at the end where you can easily see all the output. Yeah, because it's part of a pipeline, it uses a nice configuration file where you can set all your parameters in one place, and it maintains a standard directory structure so that you can know where your outputs are always going to be, same place every time, named the same way. And that also allows you to do programmatic comparison where you're taking outputs from multiple runs and combining them. So there's a lot of cool stuff with this pipeline. In addition, it's containerized, which means it's provided in a Docker container. And what this allows you to do is run the software on any platform, whether it's a Mac or a PC or Linux or a cluster, and it will run the same way because it's like a virtual machine running inside this container. So this is available as a preprint now, and should be coming out in GigaScience, we hope, in a few months. Another tool we have is called Mito Helper, and this my postdoc Gene developed. And what this does is it's designed to work especially with MitoFish, which is a database of mitochondrial sequences of fish. And it's a really nice database, but there's some issues with it. And one is that it's not always clear which mitochondrial sequences, which regions they correspond to, right? Because the mitochondrial genome is huge. So some of the sequences might be whole mitochondrial genomes. Some might be just the 12S gene, maybe just the 16S gene, maybe just the CO1 gene. And so you can use this tool to pull out just the sequences that are for the region that you want. And you can also align them to a reference to see like which part of the region is there, and also like do kind of things like get properly formatted records and connected with NCBI type taxonomy and all sorts of things. So this is a useful tool for anyone using mitochondrial sequences, especially to study fish. And so that's a brief summary of some of the tools that we're developing. And now, so I want to talk about how we're applying these to our research. So I showed earlier this study where we were testing different extraction methods with those Stereovex filters, but I didn't say too much about like where we got the samples or what we found in them. So AOML, our lab, is here in the center, and then across the street and on the water is the University of Miami Rosenstiel School. So Rasmus is the abbreviation, and so they have a dock there that we sampled from. So we just dropped one of those Niskin bottles into the water, closed it, brought it back to the lab, filtered it, and then did the extractions. And so we were curious to see, what kind of fish could we detect in the eDNA from that site. So when Sean, my postdoc, did the analysis, we found lots of fish, but what was interesting was found some fish that would be expected to be there and some not. And we weren't, neither of us are fish biologists, and both of us are fairly new to the area. So we don't necessarily know what you would expect to see in those waters or not. So we actually we sent out a survey to anyone who we could think of who would be familiar, whether fishermen or marine biologists from the area, or et cetera. And we had asked them to take the survey and say, listing these different families, would you expect to see these fishes in Bear Cut, which is the name of that region of water? And so this is an example here. So, yes, no, not sure, yes, but I've never seen it. I just think it should be there. It makes sense that it would be there, that kind of thing. And this was because we wanted to know like how accurate, were we? And in general, most of the stuff that we found, people thought, made sense that it was there, but there was a few exceptions. In particular, highlighted here is Oncorhynchus micus, which is rainbow trout, Clupia Harengus, and Asmeridae. So these are herring and smelt. And so rainbow trout, herring, and smelt, and these would not necessarily be expected there. And so we thought about this, and some of the responses to the survey, people were like, you might check the Miami Seaquarium, because I believe they can feed some of their animals those fish. And so it turns out we talked to the curator there, and he said, yeah, so like every day we take the fish guts that were left over from feeding our marine mammals and so forth, and we dump them on the side of the water, which is kind of in the lower right of the screen here. So they were dumping these exact species that we were detecting: rainbow trout, Atlantic herring, and smelts or capelins, and dumping the guts in the water. And that's just around the corner from where we were sampling. So it seems highly likely that what we were detecting was these fish guts. And that was actually really, really comforting that we could detect this signal, and it made sense. And even though it wasn't a natural signal, it was natural in the sense of it was there. We were detecting something that was most likely there. So moving on to another study, this is more getting out into the ocean and sampling. So this is called the CMAP survey, so the Southeast Area Monitoring and Assessment Program. And in particular, this is called the CMAP Reef Fish Video Survey. So what our collaborators at the Southeast Fisheries Science Center, which has its bases in Miami, but then there's other labs in Galveston, Texas, and in southern Mississippi, Gulfport, Mississippi, or Pascagoula, I think is their main lab in Mississippi. In any case, what our collaborators are doing is they've built this device, which is a camera trap that has, they put some fish bait on there, and then it also has these Niskin bottles that you've seen before, but now they're mounted on this this cage. And so the idea here is we're going to put this thing down in the water with the bait. Fish are going to come eat the bait. We'll get video record of them, and also we can, at some point in time, we can close those bottles and get a water sample that we can measure the eDNA in there, and ideally be able to see how closely the eDNA matches the fish that we observe visually. And this includes commercially important taxa like red snapper, and we're also interested in more generally the community there. So we're going to look at microbial and plankton sequences as well. So today, we just have data so far from our 2019 cruise. And we have, in this case, it was done a little differently. The water was actually from a CTV, this device that has the bottles mounted on the upper right here. And, but in 2021, which we're currently analyzing now, we'll have that sort of more traditional or optimized camera trap design. So we've got stations here from Key West, Tampa area, Panama City, Galveston, and Brownsville. And these are pretty shallow areas, so most of them are less than 100 meters in depth on the shelf. And so most of the data we have that we can make sense of so far is the bacterial data. So that's mostly what I'm going to share today. But there's some cool patterns that came out of this. So there's really clear delineation between the surface and the bottom samples. So on the right here, the richness, which is the number of unique sequences we find, is clearly higher in the bottom than in the surface samples. And that's not unexpected. We tend to see that a lot. And there's also clear differentiation in what we call beta diversity on the left. So this is how different each sample is from the other. Clear separation between the surface and the bottom samples. But there's more to it than that, because we can actually explain a lot of the variation, not just with depth, but with region. So if we take, this is a somewhat different analysis where we're, it's called redundancy analysis, where we're trying to maximize the variation or the separation of the samples based on environmental parameters such as oxygen, fluorescence, which is a proxy for chlorophyll, transmission, temperature, salinity, these kind of things. And so we see pretty good separation of the different regions based on their microbial communities. So especially with most of these lower left samples from the Panama City area being more different than the other 3 regions, and also some structuring of those other 3 regions, and with things like oxygen and temperature and salinity driving those patterns. So this is going to be very interesting to compare with the fish data, because potentially we're seeing differences in the microbial community from linked to differences in the environmental parameter space, and then can build that into models of fishery fish abundance based on those patterns. So when we look at the fish data, this is still an early glimpse. There isn't a lot of structuring based on surface versus bottom, but there's a lot more in there, and we're still working through that data. So that's definitely a work in progress, and something that we'll have to report back when we have all the fish data especially from both years. So I wanted to move on to another project, and this is close to you guys here in Lake Erie. I believe this is Michigan here on the left. And so what this project is we're trying to study sort of 2 things. One is harmful algal blooms in the Great Lakes. In particular, microcystis is a toxic cyanobacteria. And then we're also developing and testing a new technology, which is this uncrewed system type of thing, an autonomous underwater vehicle, which is AUV, and then it's an LR AUV because it's long range, and then it's linked to a 3rd generation environmental sample processor. So in simple terms, what this thing is, it can swim around in the water and collect water, filter it onto a 5 micron or 0.2 micron filter. And then from that, we can extract that DNA in the lab and then study what was in that sample, either by qPCR, amplicon sequencing, or metagenomics. And so one of the things we did was just to study how good is that device, that autonomous sampler. This here, it's called 3G ESP in the graphics. How good can that replicate the traditional hand sampling? Ideally, we would get more or less the same answer using these 2 different technologies. So looking at here, it's Shannon diversity of the 16S data from the 5 micron filter, 16S data from the 0.2 micron filter. They're very similar. And then metagenomic sequencing from the 5 micron and 0.2 micron filters. The only case where there is more of a difference between the 2 technologies is with the 5 micron filter of the metagenomics. So for the most part, this is a technology that can capture the patterns that we would see with traditional hand sampling. And so some of the things that we can then see is this is just looking at the qPCR data. We've got lots of other data from the metabarcoding or amplicon sequencing, the metagenomics, and actually some toxin detection technology as well. But what we can see is that this device does a really good job of capturing patterns in cyanobacterial abundance, which is shown on the right. This is total cyanobacteria from qPCR in 2018 and 2019. In 2019, there was a much stronger bloom. So those those points are going to be darker and bigger in 2019. And similarly, the quantitative PCR of one of the toxin genes, one of the genes that makes the toxin that makes these cyanobacteria so toxic is MCYE. That's also higher in 2019. You can see how it varies between different stations. And this, we have other data showing that this corresponds really well with things like flow cytometry and metabarcoding and metagenomic data. So now I'll turn to our sort of plans for more global eDNA studies, and this is still in the early stages, so it's more of a plan than any data so far. But we are part of a project called BioGoShip, which is built on the GoShip project, which is a global ocean sampling set of expeditions. So what you see in this map is around 66 different lines. I'm not sure all of them are shown here, but these are sampling lines that NOAA ships and then our university partners from around the world really are going on over a decadal scale. So over a 10-year period, if everything goes well, we will have sampled each of these lines at one point. There's been some setbacks due to COVID-19 and so forth, but in general, that's how often these cruises are going. So it's a really great opportunity to have a long-term time series on a global scale and to apply some of these technologies that I've been talking about, specifically with eDNA and omics, to get a deeper understanding of the microbial communities. And so we're looking at especially plankton specifically. So what are, looking at global patterns of taxonomic and functional diversity, how physical processes affect these patterns, how plankton might affect global cycles of carbon, nitrogen, and phosphorus, how they might affect carbon export fluxes and how those vary, and also just looking at global patterns of plankton size spectrum with latitude, with depth, with different nutrient regimes and so forth. And so there's a lot of different methods that go into this. It's not just omics, but it's also various optical methods, imaging methods, flow cytometry, etc. So this is going to be a really exciting new focus for us, and so we're playing a part in particular the omics, but also in integrating all these different data types. So this link here is to a sort of commentary that we published where we're describing the plan for this project. And so I want to move on now and talk about kind of a different style of project. This is a more focused on a host microbe symbiosis in the Gulf of Mexico, and this is something called the methane ice worm. So this is taken from a presentation that Jean Lim, my postdoc, gave at the Ocean Sciences meeting earlier this year. And her talk was called Microbial Communities Associated with Cerso methanicola, colonizing a methane hydrate in the Gulf of Mexico. So methane hydrates are these geological formations that occur where there's methane seeps. And basically the methane reacts with the water at pressure, and it forms this methane clathrate, or methane hydrate, and it's what's called colloquially like methane ice. And so this methane ice forms at the bottom of the sea, and large amounts of it stay there because of the pressure that's holding it there. So it creates this really cool environment, and of course microbes and and even higher organisms have adapted to consume the methane and sulfate that's coming out of that environment. So the only, as far as we know, the only macrofauna that live on these methane hydrates are this methane ice worm, the Cercoe methanicola, which is a polychaete worm. And so if you're lucky enough to find one of these methane hydrates in the bottom of the Gulf of Mexico, this was taken about a little over 500 meters depth. So, you know, almost half a mile or so below the surface. It doesn't look like much from a distance, but if you zoom in, you can see the methane ice is this kind of yellowish white stuff. And in these little holes are all these polychaete worms that are making, you know, living there, making their life there. And this is what they look like up close. So it's not a gutless worm. It has a functional gut. It's thought to be a bacterivore, so it eats other bacteria. And as far as we know, there's no particular intracellular symbionts or epibionts, but this is really not a very well studied organism. So in 2009, these samples were collected using a submarine. And it's off the coast of Louisiana at about a depth of 543 meters. And so these, there was a handful of worms, I don't know, maybe less than 10. And we ended up, this was before my time, but what the researchers did was they were able to separate out some gut contents and also some of the tissue contents of the worm. And so we ended up with basically 3 metagenomes, so 2 from the gut. One was sequenced with HiSeq, and that's mainly what we'll talk about today. There was some HiSeq data, but it wasn't very substantial. And then another metagenome from the worm tissue. So this is the W, and G is gut, also sequenced with Illumina HiSeq. So it's a very we don't have a lot of replication. We just have one metagenome from the gut and one from the worm, and there's a little bit of overlap in the DNA or the material that was between those 2 samples because the surgery wasn't 100%, you know, exact, but was the best that could be done on a ship in the middle of the ocean. So looking at the gut library, it's predominantly Campylobacteria, and these are sulfate reducing bacteria, and there was some other stuff there too, but that's by far the predominant one. And there's some details about how we did the analysis of using 16S data from within the metagenomes. The most abundant, by far, as I said, was Campylobacteria, or Campylobacteriales, the order. Within that, they were assigned to Sulfospirilleaceae family, and within that, Sulfospirillum genus. And so, but there was around 16 MAGs, or metagenome assembled genomes, that were recovered. These are also called bins when you assemble the data and then bin them into MAGs. So we had, as you can see, Campylobacteria, Desulfobacteria, Gamma Proteobacteria, Alpha Proteobacteria, SAR-324, and Bacillus, or bacilli. Again, the most abundant was the Sulfospirillum, but there was also Desulfobacteriales and a few other players. So some cool diversity, a really interesting community, and there's a lot of metabolic potential in this community. So this includes sulfur cycling, including sulfate reduction, and sort of syntropic sulfur cycling, where the sulfate reduction is linked to oxidation of acetate. oxidative phosphorylation, nitrogen cycling, and a little bit of other kind of stuff like hydrogen oxidation and arsenate reduction. And so this box here on the right kind of summarizes the players that we found. So this sulfospirillum is a facultatively anaerobic sulfur reducer. Sulfur ammonis is a nitrate reducing sulfur oxidizing bacterium. Sulfur ovaceae, the sulfobacteriales, is a sulfate reducing bacterium, and then some other sulfate reducing bacteria of the disulfobacteriales as well as a Cowellia species. So a lot of detail here, but there's some really cool metabolism in the gut of these methane ice worms from different pathways of sulfur acetate and carbon metabolism, as well as nitrogen and hydrogen. And then this is even a busier slide, but each of these numbers represents the bin, and they're colored by taxonomy shown in the lower left. So there's a lot. This is like an overview of all the different carbon metabolism that's found in these different genomes from the gut. So a lot of different pathways for acetate metabolism, B vitamin cycling, carbon cycling, including pentose phosphate pathway, gluconeogenesis, reverse glycolysis, fermentation, and carbohydrate breakdown. In addition, Jean found some gene clusters corresponding to alkane degradation. So at the top here is a reference genome, and then these desulfobacteriales have large parts of operons that are predicted to be alkane degradation. And one of the MAGs, one of the genomes belonging to sulfur ammonis, has a large amount of a gene cluster for aromatic compound degradation. So it appears that in addition to all of this sulfur-based metabolism and carbon metabolism of the microbiome, that there may be some degradation capability of hydrocarbons as well, which is interesting for things like bioremediation and just interesting in general, and maybe predictable given these guys are surrounded by hydrocarbons. The gut content also has almost a complete vitamin B12 biosynthesis pathway. And then Jean also looked at the worm fragments. So this was from the other library, from the worm metagenome. And it's largely opisthocont, which is just metazoa and fungi. And she's in the process of annotating and publishing the mitochondrial genome from the worm, which will be a really great resource. And so one of the things that she found in the metagenome of the worm, metagenome of the worm, is there's cytochrome P450 clusters, which are have some similarity, around like pretty good, like similarity around 50% when you cluster them like this, or even a little bit greater, to the Capitella telata cytochrome P450. So this is a worm that's more deeply studied, and has this cytochrome P450 that may be degrading hydrocarbons as well. So it looks like the worm may have some hydrocarbon degrading potential as well. And this is, we would need more samples. So we're hoping to get back out there and sample some more worms and get eventually get a more complete genome of the host. So yeah, to summarize Jean's work on this methane ice worm, the metagenome, or the gut microbiome, was dominated by sulfur-reducing sulfospirillum, and the worm may be able to obtain things like carbon, nitrogen, sulfur, and B vitamins from its gut microbiome. And the P450 annotation suggests that Cercoia methanicola may respond or even detoxify polycyclic aromatic hydrocarbons based on its genome. And so finally, I just want to talk about briefly some of our planned work on coral reefs using some of these same tools we've been talking about. And so there's a number of projects in South Florida where they are doing coral reef restoration. So this involves things like removing seagrass and especially outplanting corals, like shown in the middle, upper middle pane. And so what we want to do is use a variety of methods, and some of these are already happening, visual observations, but also potentially add cameras and hydrophones, and in particular, DNA sampling to study how these communities come back. Because we know that if you outplant corals of a certain type that are maybe more resistant to disease or resistant to grazing by parrotfish and those kind of things, that you can get coral reefs to come back. And Florida's been pretty hard hit by loss of coral reefs. So we want to apply eDNA to these studies and give another dimension to the community response and recovery in these environments. And so we are in the process of forming proposals. Nothing is set yet, but we've got a few different sites shown here. So we've got a collaborator at University of Miami who's doing reef restoration in Miami-Dade County, so very close to our lab. You can get there in a less than an hour in most cases. We've got another collaborator at Florida International University who's doing work in Puerto Rico, and similar kind of thing where they've got some reefs that have been restored, that are being restored, and others that are not, and then they're doing comparisons. And so the idea would be to add environmental DNA monitoring to supplement the kinds of observations they're doing to detect recovery. And then we've got another site. It's pretty cool. This is even closer. This is in the Port of Miami, so right by the cruise terminal, very close to our lab, is a site called the Coral City Camera, and there's a live feed there. I'm going to see if no, I can't play it, but oh, I lost my slides. There's a live feed there, and I can share the link perhaps, but it's such a cool site. It's a webcam that's set up. I was just looking at it this morning and I saw a squid like swim by, and you'll see sharks. And so we want to set up some eDNA sampling there. And by the way, all this would be done using that SASE, that little robotic device that I showed at the beginning, to monitor what's there and try to correlate that with the camera data that's being collected there. So a lot of the different opportunities to use eDNA to study these environments and over time and over restoration. So that's all I have. I wanted to thank the different investigators that I've been working with: Kelly Goodwin, Chris Kelble, Ian Enox, Patty Roselle, all at NOAA. And then my team, Sean Anderson, Jean Lim, and then work closely also with Nastasia Patton and Paul Daniel. And then I'd like to thank the funding agencies, NOAA Office of Oceanic and Atmospheric Research, which is the big line office that we work under, and they support the Northern Gulf Institute, which funds me, as well as the Global Ocean Monitoring and Observing and the Ocean Acidification Program. And also our collaborators at the Monterey Bay Aquarium Research Institute, who developed the long-range AUV that I showed. So with that, I'd be happy to take any questions, and I'll turn off my slides and turn them back on if there's any questions that require it.
It's great, Luke. Thanks. I was thinking maybe during the questions you could try to put up that feed if it's
I definitely will.
Trouble.
I will.
It's pretty cool. I've been to it before.
Oh, you have? Good. Yeah.
But if it's too much trouble, don't worry about it. But you can send the link in the chat or something.
Yeah, let me pull it up.
I don't see any questions in the chat, but if people want to raise their hand on their emoji thingy, Dr. Arnosti has a question.
Thank you, Dr. Quinn. fascinating talk. Really, thank you for visiting us. We wish it were Miami weather. We're hoping to get it soon. One of the things that comes to mind looking at this environmental survey is that you guys must be talking about and working with integrating it into all the physical oceanography, the pH, temperature, streams and stuff like that. Is that done, like, how do you do that? And do you do it inside of NOAA? Do you do it as a collaboration worldwide? And what kind of computational challenges do you have when you start merging bio data sets with the physical data sets?
Yeah, that's a great question, David, and I'm happy to be here for sure, even virtually. So yeah, so we do that kind of integration, and one of the cool things about NOAA and AOML is we have people that do that kind of physical and chemical oceanography in our building, and so we can, when we're more back in the lab, we can just walk down the hall, or now we can call somebody up. But so that data is all collected on the cruises. Standard stuff is things like salinity and temperature and chlorophyll and oxygen are always collected, and then you can do nutrient measurements as well. We're also working on integrating satellite measurements. That's I think one of the exciting new directions, and we've got a new guy in our department whose his focus is on remote sensing and satellite data. So, but yeah, the data challenge is definitely there. We've been working on data management strategies, so how we can all sort of, we're not all going to use the same standards necessarily, but at least within a given field we can try, and so that will enable us to have better ability to integrate data sets, if we're talking the same language and familiar with each other's standards, and then where the data are shared, how to access them. And so that's definitely something we're doing. And then my boss actually is an expert on sort of ecosystem modeling and ecosystem-based fisheries management in particular. And so we're definitely thinking about how we can integrate these different data types into a single model that can predict different things. And there's a lot of different statistics that goes into it, and that's where some of the machine learning comes in that I didn't talk too much about, but we're using different machine learning algorithms and some cloud computing, which enables that, and figuring out where we want to do that computing and stuff. So it's it's definitely I think we're well situated to do it, but we're still in the early days of really integrating everything.
We have another question from Bartolomeo. Sorry.
Hello. Hi. Yes, I am Bart from the Fish Pathobiology and Immunology Lab of MSU. And I would like to ask you if you may provide any example of how your research results have been applied to design management solution, or maybe policies that may be implemented by environmental agency or government or any other.
Yeah, good question.
Thanks.
So to be honest, that is an area where I am still more in the research realm. So I'm trying to think of a good example for you of where some of these research findings have made it to policy. I would say, well, I can give you a good one. So right now, there's a disease called stony coral tissue loss disease that is really ravaging coral reefs, especially in the Miami area. And so our researchers in our division are studying how the transmission of that occurs, whether it's via ballast water of ships, or it could be via sediment or some other mechanism, and how you might say treat the ballast water with UV or those kind of things to abate that issue. And so there's definitely a lot of conversations going on between NOAA and different organizations to figure out how best to prevent the spread of this disease. Another one that's very commonly done. So there's a couple of congressional bills that we have to, that our research supports, which is, one is the Endangered Species Act, and the other is the Magnuson-Stevenson Act, which is basically about fisheries conservation, was related to the sardine collapse of the 1930s or whatever it was. So we do things like, or NOAA Fisheries does things like stock assessments and so forth to say, they don't make policy decisions, but they provide the data for those policy decisions about fishing, overfishing, how much fish can be taken in a given year of different kinds. And so the kind of things we're doing right now, eDNA data doesn't go into those stock assessments, but we're hoping that in some point it will, that we can provide data that will be used for those fisheries decisions. Because if we can do the kind of sampling that we're doing, especially with these like remote operated vehicles, that could be a lot cheaper than sending out a big ship to do a survey. And it's also less destructive because we don't have to trawl and haul in fish and plankton and so forth. If you could get a good estimate just from the DNA in the water, that would be much less destructive, potentially cheaper, and so forth. So yeah, there's a lot of links to policy. We're the part of NOAA that's more research focused, but it still has links to policy, and ideally the research we do will have some kind of transition into an operational activity at some point. So it's definitely more applied, for like maybe the students in the audience thinking about like a government career versus an academic career in science. It's definitely more applied in NOAA, but that can be exciting too, that to have sort of know that your work has potential to make a big difference in conservation or in economy or whatever it might be.
That's great, Luke. You really kind of led into my question, but we have one from Tommy Vogt.
Hi, Luke. So DNA is notoriously very stable. And so you're probably going to pick up DNA from organisms that are currently there and organisms that were there at some point in the very far past that they're not there anymore. And so when you're picking all of these 8 DNAs, how do you separate those things out?
Yeah, that's a great question. In some cases, we have studied, or our collaborators have studied half-lives of DNA, so we have an idea of how long DNA can stick around, and it tends to be on the order of days, hours to days in these environments. It does depend on things like temperature, oxygen, pH. Generally, especially like higher temperature or higher oxygen will cause the DNA to degrade quicker. So we have to keep that in mind, but it's still a challenge, and this is something we're thinking about with those, especially with those coral reef studies where we have maybe the spatial resolution is things are closer together, and water is a liquid, right? So it can flow from one site to the other, and so we got to keep in mind those currents, those half-lives. So we would love to have idea about the half-life of any given. We'd love to have detailed information about like individual species, and also like the different fractions. So whether it's free DNA or in cells or mucus or feces or whatever it might be, how those different fractions are degrading and under what conditions. We just don't have too much of that data right now. So I think your question is super valid. It's something we're we don't know a lot about. We're kind of have to build that in, that kind of uncertainty into the interpretation.
Thanks.
All right, well, a little afternoon, so lunchtime for Luke and all the students. Want to thank you for your great talk, and it was nice to hear some kind of different stuff. We even had some people attend, I think, from different departments, so that was cool. Here's
Good.
the stream.
Can you see the, I'm just trying to show the camera.